My team's very much interested in understanding the fundamental biology of muscle wasting disorders, and so that could range from genetic conditions such as congenital muscular dystrophy, or also conditions such as aging. We're interested in understanding, why does this disease occur? How does it occur? And importantly, can we then subsequently translate all this knowledge into improved prognosis, diagnostics and therapies for these conditions? One of the expertise that we have is in using fish models to study congenital muscular dystrophies, and importantly, these are the zebrafish. Quite often when I present this, I get a question, how can fish actually model muscle diseases? Fish have muscle structure that's very, very similar to humans, and so what we learn from fish can actually be subsequently translated into humans. But beyond that, they also have a range of different advantages. For example, fish offer prolific reproduction, so one male and one female can give hundreds of embryos. These embryos are actually transparent, and so we can label our structures and areas of interest with different colors. I can visualize the process happening in real time. These embryos are externally fertilized, and what that means is that we can go in and genetically manipulate the cell. We can add a gene or remove a gene, and then look at the consequences of this modification on disease manifestation. Additionally, zebrafish also develop very rapidly. So a disease that will take about 6 years in a patient to manifest, but in a zebrafish it can take as early as 2 to 3 days. So this is really advantageous when we're trying to model muscle diseases. And finally, not forgetting that fish actually live in water, so we can add all sorts of drugs and chemicals into the water and look at how these are impacting the disease state itself. So we can test a range of different therapies with the goal of identifying therapies for congenital muscular dystrophy.